Um, Hi, my name is Dylan Kudva. I'm from Khan Lab School.
My name is Aldo Salanya. I'm from Alsa High School.
And I'm Francesca Tom, and I attend Skyline High School.
And we're doing the Younger Lagoon Reserve Project for environmental science.
So here's our presentation outline. First, we're going to go into our background information. Then we'll go into our research question, our hypotheses, our study site, and our methods in the lab, our results in the works for us, and then we'll go into a discussion and what's next for us.
So restoration is when native species are planted and invasive species are controlled or eliminated to recover an old ecosystem. Restoration can help with ecosystems because native species might have better carbon sequestration. And in the picture on the bottom corner, a restoration attempt was made, but as you can see, both sides look the same because there wasn't enough research done. And another reason restoration is good is because native species provide more ecosystem services.
So as climate change continues to affect our ecosystem and affect several species of plants around us, much of the variability of restoration will be affected because of the environmental filters of these existing native plants. As we could feel on our own, California continues to grow in temperature. However, we are unsure about the future of precipitation here. We do anticipate more extreme weather patterns, such as more flooding and droughts. Species interactions is a process of when many restoration practitioners, they will often not go into depth about the research that they're doing, and they'll often go into a land and start to plant these native species that they have, but due to underfunding and the lack of research, they end up affecting the local ecosystem of where the plant was planted. So species interactions and environmental filters go into affecting response traits and effect traits. Response traits are traits that get directly affected from the species, so that includes a bigger leaf area or a mass change in the leaf. And effect traits is what happens after to the response traits. So for example, that would be due to the bigger leaf area in a plant of a leaf, they will end up having more shade and make it more competitive for the species around it, for other animals and insects, and that will end up affecting the ecosystem around that plant.
A functional trait is anything that can be categorized or measured in a leaf. So this can include leaf thickness, which can mean the leaf is well insulated, or that it needs more photosynthesizing. If a plant has low specific leaf area, which represents leaf overweight, the plant is more likely to have lower nutrient concentrations, lower rates of respiration, and low rates of photosynthesis, increasing the chance of drought resistance. We also look at the major vein length, the ratio of the vein length to the area. A high VLA suggests drought tolerance, and we look at the carbon to nitrogen ratio. A high carbon to nitrogen ratio also suggests drought tolerance. And finally, we look at the carbon isotope 13, which determines water use efficiency. The more there is, the more resistant.
So phylogenetics are evolutionary relationships determined by similarities between species. The farther apart two species are on a phylogenetic tree, the less similarities they have. Cumulative phylogenetic distance, or CPD, is the distance from one species to all of the species. And if a species has a greater CPD, so if they're less related to the community, that might provide an advantage for them because they might not have the same problems that the other species do. So our research questions were, how can functional traits affect survivorship in droughts? How does distance in phylogenetic relationships affect survival? And do native plants react differently than non-native plants to treatment?
So the hypotheses that we drew in our research were, as drought occurs, invasive species will do more poorly than native species, which can be seen through the functional traits of the plants. And the second one was species with functional traits that allow them to be smaller will use less water and therefore be more drought resistant and have greater survivorship. So here's our study site. It was on the coastal campus of UC Santa Cruz. To test these hypotheses, we designed 3 different types of plots where they were each planted with 12 different native species, all with the same pattern. This was done to give the different ecosystem for each plot. All of the plots were mowed in the beginning and then weeded twice in the first year. So to the left, that's the shelter plot, and that was designed to create a 1 in 100 year drought, and that's designed to create to limit the amount of water that gets in. So 60% of rainfall would not get into the plants at all, and so that was designed for the drought. And to the right that's the control and water plots. They look very similar. The control plot has nothing done to it, so it has nothing done. And for the water plot, it was watered 2 liters in the first week, once a week for 2 months. And that was designed to show how many restoration practitioners, they'll often go into the land and begin to start the restoration, but then due to underfunding, they're not able to follow through with the process. And so these are the 12 native species that we did. They were planted in January 2016, and they were chosen based on the restrictions required by local genetics. So, which means that they were chosen in a small distance away from where the planting site was. There were different types of shrubs, grasses, and forbs, which is the flower.
So methods in the field. Starting in 2016, we measured survival and growth. And then in 2018, we began collecting the functional leaf traits for the lab. In 2019, we replanted 3 dead species and took traits from the other surviving species. And we also took traits from 3 implanted native and 9 non-native species. So in the lab, we scanned leaves while they're still fresh to retain their size. And then we weighed their dry mass, and this is so that the amount of water in each leaf would be standard across all species. We found the leaf thickness with a micrometer. We ground leaves for carbon and nitrogen isotopes by the plot, and then we used a mache to find the leaf area and major vein length.
So these were our findings, and each point on here represents a plot, and each plot represents the plant community within the plot. And the further apart the plots are, the less similarity they have to each other. And the important thing to take away is that the drought treatment actually affected the plants while the water didn't, because the water and control treatments are overlapping, but the drought isn't overlapping with the control.
So here's another table that we made for the main. So everything to the left of the plot of the table was functional traits, except for the main treatment, which is controlled drought and early water. And just to know, there's a no axis at 1, and that means that if it lands on there, that there was no change to the plot, nothing happened. And if anything falls to the right, it increases the risk of death, and if anything falls to the left, the decrease in risk, there would be a decrease in the risk of death. And so we found that the overall size and growth rate and the major vein length per unit area, they would both fall to the right of the graph, which would increase the risk of death of the plant. So the bigger they would get, the more risk of death they would have. And then for the CPD, the greater distance in CPD, the death risk decreased. So in other words, the less related these plants were, the longer they would survive. So in the works for us, we're still currently processing traits for all the species. This will lead to us figuring out whether or not invasive species determine plant community composition. And this will also help us determine whether or not some traits are stable by family, and if other plant-specific species will help others thrive in those certain conditions. We're also still processing carbon isotope samples and nitrogen isotope samples.
So the native species might have done better in the water plots because they could be watered in their early stages of development, which allowed them to grow more. And they also might have done better in the drought plots because the invasive species weren't adapted to droughts, so the native species could grow more in their early stages and then have a competitive advantage later on. And species with a greater CPD might have had a greater advantage. So like species less related to the community might have had a greater advantage because they have different ways of accessing food or water.
Management implications. Drought affects plant community composition, so restoration practitioners might have to adjust these practices. The younger lagoon the site where our project is being conducted, mows to control the invasive species. And as Jillian previously said, plant communities are changing to those with deeper roots. So this method may no longer be effective since plants with deeper roots are able to sprout after mowing.
So these are some questions we have for the future. So one of the questions is this pattern true for all types of grasslands, and could it be true for even other habitats in the same type of climate? So this could help see if this could work on a larger scale, so not just grasslands. And also, how are reproductive traits affected by these treatments? Because we looked at leaf traits, and if we looked at reproductive traits, we could see if this could work for a long-term situation.
We'd like to acknowledge our mentor Justin Lung, Dr. Michael Loik, Dr. Karen Hall, and Dr. Wei-Shing Chen, and also the SIP program and our families for their unconditional support. We'd now like to open it up for any questions. Thank you.